I'm very excited to share about the work that we've been doing, that how to turn big biology data into insights on disease using the power of circulating biomarkers. I want to emphasize that this is really a team effort. There's a big data science and data engineering team behind all of the work. Actually, our head of data engineering, Edmondo, is sitting in the front row. So if you have any difficult technical questions, you can save it for the break session. So first, let's take a step back to understand some of the pitfalls in the drug development today that we are working to overcome. Despite many advances in drug development, more than 90% of the drugs still fail. And what's more, even for the drugs that have been FDA approved, it only works in a small proportion of individuals. And in fact, if we look at a recent review of 85 cancer drugs that are FDA approved, and we find that the median response rate to these drugs, it's only 41%. So that's actually pretty low, and I don't know if it's surprising or not to you. But it actually, all these issues is because of the fundamental challenge here that a large portion of the patients just don't actually respond to the drug. And so it's not surprising or it's expected that the response rate will be low. And this would cause lots of adverse consequences. For example, it could increase the risk to patients, and it could cause time delays for the trial of drug development, and eventually accumulate expenses for the trial. And much of this challenge is actually stemming from a fundamental mismatch of how we define diseases versus how we develop drug. And this mismatch occurs because diseases are singularly defined by the end outcome or the pathology that is manifested. But in fact, most diseases likely represent different mechanisms and different pathways that resulted in a shared pathology or the manifestation or symptoms. And common diseases such as diabetes or Alzheimer's disease, for example, can be very heterogeneous in its origin. As drugs are typically developed to target a particular mechanism or pathway, it would not be surprising that only a portion of the patients would actually respond to the drug. And this disease heterogeneity creates a challenge for, one, drug efficacy, because it leads to overtreatment of populations that are non-responders to the drug. And two, drug safety, as those non-responders absorb all the risks of taking the drug without any benefit, while at the same time with their disease progress unchecked. And three, increase the costs for the drug development, because the reason that we have to enroll more patients now to get enough responders to prove the efficacy of the drug. So how can we overcome this challenge? With biomarkers, especially the biomarkers that are circulating in our blood. Fundamentally, biomarkers allow us to peer into individual and align the specific patients with the specific disease state, for example, the mechanism of disease and the specific drug. Using biomarkers, we can theoretically identify patients whose diseases are caused by a particular pathway that the drug is actually targeting, and therefore increase their chance of response to the drug. And there's actually hard data to suggest that this is more than just theoretical. It is in fact the case. An independent retrospective analysis across over 20,000 clinical trials from the last several decades shows that the drugs that developed with the biomarker have 2 to 10 times increase in FDA approval rates. So not only the approval rate is higher, but also the time to approval is 20% faster. Overall, clinical trials that use biomarkers are way more efficient and faster. So to date, much of the focus on clinical biomarkers have been in the genomic space. While genomic biomarkers have had a profound influence, especially around the different types of cancers, it is clear that the underlying genetic predisposition can only account for a minority fraction of the risks that most human diseases have. A broad-scale analysis across most adult-onset diseases shows that for the majority of the diseases, as you can see, all the ones that listed in the bottom, have only less than 30% of their populational attributable risk due to genetics. And even the most inheritable diseases, like the two autoimmune diseases, asthma and steroid autoimmunity, have just over 50% of their population attributable risks due to genetics. So that means the vast majority, more than 70% of human disease risks, is not captured in the human genome. Because the genetics is set at the moment of conception, the moment when we were born. But everything that occurs after birth, like where we live, what we eat and drink, and how much we exercise, etc., is not encoded in the human genome. And all these factors can have tremendous influence on health and disease. Yet, the vast majority, and more than 85% of all these environmental and the lifestyle exposures and the corresponding biomarkers for these diseases are still unexplored. This is the great opportunity for small molecule biomarkers. Small molecule biomarkers can read out both genetic and environmental factors that underlie human diseases. And most importantly, given the size and the chemistry of these small molecule biomarkers, they can be released from peripheral tissues and tumors and readily cross the cellular membranes and other biological barriers and enter into central circulation. And these biomarkers or molecules can be read out at a very early time point in the disease progression. And since we can measure these biomarkers in a non-invasive way, so we can do this more frequently and be able to get the early and the real-time disease process readout. The small molecule biomarkers can inform us on the host organ level physiology, local disease processes, and the effects of exposures on the individual stemming from both genetics and the environment. Human blood contains thousands of these small molecule biomarkers. And as mentioned, more than 85% are still unmapped. This is at the bottom of the iceberg. A key reason has been the bioanalytical technologies that limit the speed at which we can achieve the analytical scale. And this is the challenge that Sapient has set out to tackle. And next, I will describe how we achieve the small molecule biomarker discovery at a high-throughput scale. Sapient represents a discovery engine for small molecule biomarkers and operates foundationally on these three core components: analytical technology, biocomputational integration, and a human biology database. I will go through these components in the next 3 slides. The first is the next generation mass spectrometry technology that allow for measurement of more than 11,000 small molecule biomarkers in each blood sample. And we handle this analysis at unprecedented speed with less than 1 minute per sample and more than thousands of samples per day. Our computational component includes statistical and machine learning tools that we use to integrate our mass spectrometry spectral data with various high-dimensional biological data. And I want to emphasize that we are able to extract and interpret this high-volume data with our proprietary mass spectrometry data extraction pipeline. This pipeline uses a neural network-based pattern recognition and a distributed image processing infrastructure. Our machine learning algorithms can remove up to 90% of the false spectral peaks without reducing the true signals that are manually labeled by human experts. So it's essentially an automated, scalable tool to replace the human experts. And the third component is our human biology database that encompasses data that have been generated using our technologies out of the tens of thousands of blood samples collected from individuals around the world. And there are over 7 million matching phenotypic data points for these blood samples, including cohorts across diverse backgrounds, demographics, geographies, lifestyle, diseases, and medication regimens. And decades of follow-up across patients with data on diet, exercise, mental health, and clinical outcomes. And this is very important because we want to be able to use the baseline blood sample biomarkers to predict disease incidences and outcomes. And as well as the genetic information for a subset of the subjects and the microbiome sequencing data so that we can connect the small molecule biomarkers with underlying genetics and exposure from the gut microbiome. All of these components are embedded into an integrated discovery platform, which you can see here. From raw data generated with the high-throughput technologies to data ETL from on-premise to AWS cloud, the machine learning and the distributed computing-based feature extraction for the spectral data, and finally, statistical and machine learning models in Databricks and harmonized data in the data lakehouse. All of these modules are used together to generate, extract, ingest the biomarker data, and also to integrate with other clinical data to derive insight on human health and disease. To give you an idea of what we can learn from these approaches, shown here is analysis across 50,000 diverse individuals from all around the world. And what you see on the left, this circle line diagram, is that not every individual has exactly the same molecule in the blood. As we would expect, we all eat slightly different food and live in slightly different area and are exposed to slightly different environments. So across these 50,000 individuals, we can actually measure about 180,000 circulating small molecule biomarkers across the entire population. So this is counting all the unique small molecules in these 50,000 people. And about 12,000 of these molecules, we can consider them as common because they are present in more than 50% of the populations. So they're shared by at least half of the population. And among these 12,000 small molecules, about 3,000, so that's the core, these are the universal molecules because they are present in every individual's blood, regardless of where they live and what their lifestyles are. So these 3,000 molecules are the core metabolism for human biology. This includes the common sugars, nucleotides, lipids, proteins, amino acids that are required for us to walk and talk and live in this world. So looking at the figure now on the right, we can begin to understand not only the presence or absence of these molecules, but also the dynamic range of these molecules in the population. If we, the figure on the right is only counting the 12,000 common molecules. And we find a very interesting pattern that about 1,000 molecules on the far left of this figure that are highly stable in all individuals. Like they have a percentage of a coefficient of variation less than 5% or 10%. So this includes molecules such as glucose that are very tightly controlled by the body via homeostasis. On the other hand, on the far right side of this figure, the blue region, we can see that about a few thousand molecules that are actually hypervariable across the population. Well, we all have these molecules in our blood, but their concentrations can vary by orders of magnitude. And these molecules could have incredible predictive power and can be used as the biomarkers for understanding human health and disease. Next, we want to look how these molecules change over time. So first, let's look at the relatively short period, over 24 hours. That's the orange histogram here. And certain molecules in blood can be quite dynamic over the 24 hours. And we find that more than 2/3 of the molecules change more than 50% within a day. Well, this, of course, has to do with the fasting and the feeding cycle and the sleep and the wake cycle. So chemically, as represented by all these small molecules in your blood, you are very different now at this talk from when you were last night when you were having your dinner. And so most of these molecules are not stable over 24 hours. However, if we look at a longer period of time, and now we're talking about the blue histograms. So if we take our blood samples today and measure again in 2 years, but under the fasting first morning condition, then the variations are actually little. As we can see that most of the molecules change less than 50% in this blue histogram. So this stability has to do with long-term duration of exposures. We tend to have the same habits over years, eat the same thing or live in the same area or have similar lifestyles, and we're not subject to big changes. And so this determines the stability of the fasting molecules. And this consistency and stability can really allow us to do small molecule biomarker discovery by precise quantitations. So with our data, we can also begin to ask, what are the factors that influence these molecules in our blood? Using data from several hundred pairs of identical and fraternal twins, we can statistically compute the amount of influence that comes from genetics versus the environment. And when we do so, we find that the environmental factors are absolutely key to the determinant of the molecules in human blood. As you can see, all the dots are centered around the top. That's where the environmental factors are contributing most. And this is where we begin to map what we would consider to be the non-genetic landscape of human disease. One of the environmental factors that gained most attention in recent years is the gut microbiome, which we know has a huge influence on human health. And so it is on the molecules in circulation. So as shown in the figure on the right, we find that over a third of the molecules floating around in human blood are actually produced by the gut microbiome. We also find that other dietary lifestyle exposures and some clinical variables can have profound influence on circulating molecules. Everything from age, gender, BMI, race, socioeconomic status, geographic location, blood pressure, amount of exercise, etc. All of these factors can influence circulating biomarkers. I would like to end this talk on a very high note and show a successful real-world example of how we can use the insight derived from our data to predict disease prognosis. A big challenge in clinical trial enrollment is how we can efficiently identify individuals with high risk and enroll them for the trial. So out of the tens of thousands of biomarkers measured in blood samples from the tens of thousands of individuals, we can identify key features and build a risk score model that can better predict individuals at a high risk for disease events compared to the standard of care prediction method, as shown in the figure on the left. Across different human studies, the results are very reproducible and consistent. And we find that this biomarker risk score at varying percentile in the population, as shown by the orange dots in the figure on the right here, enriches not only for disease events, meaning that they allow for less individuals to be enrolled because they're better at picking patients with higher risk, but also require fewer individuals to be screened. And this really reduces the amount of effort that is needed for a clinical trial. And you can imagine how this new risk score may greatly improve clinical trial design and make the trial much faster and also smaller in size. Now, looking across our human biology database, we can similarly determine that this biomarker risk score can really predict individuals that are of high risk for disease progression over time. This is over 15 years of time. And this is really validating its potential as a clinical trial enrichment strategy. So I hope the examples that I showed here today can help you to understand the power of circulating biomarkers and how our platform and data can be used to map the influential non-genetic landscape of human health and disease, and to convert big biology data into actionable insight. So thank you again for your time and attention, and thank you to Databricks for allowing me here to present the work, and I would be happy to take any questions that you have. Yes?
Yeah, so this must be an obvious question you've already considered, but so if you're talking about germline genomics, I agree, it's like 15% maybe in terms of how it influences, but phenotypic stuff is like for oncogenes is a huge deal. Yeah. In terms of doing genomics. So it begs the question of why don't we use both together? And wouldn't both together combined give you a much more thorough analysis?
Yeah, definitely. I totally agree. I couldn't agree more. So here we're emphasizing the small molecule biomarkers, but it doesn't mean that we're excluding all the other components. Actually, a big part of the data aggregation and integration effort is to put all these pieces of puzzle together. We are actually doing a lot of genetic data analysis as well and connecting the genetic mutations with the circulating biomarkers. So the benefit of circulating biomarkers, it not only read out the germline mutations, as you mentioned, and if any somatic mutations happened in tissue or locally, the molecules can also read out as a functional change of these mutations. And so we're really trying to use all information we can get to link these connections and eventually to find their connection to diseases.
Some of us are doing massive
studies. Yeah. So we really hope to use this platform together with all the other existing technologies, and on the data side to gather all the data together and to best understand what all these different factors contribute to disease.
Yeah, and it sounds like we need to get a mass spec.
How much data are you processing per study and per trial or per experiment, and how often does it take, or how long rather does it take to process one of those? For instance, if you were looking at a particular biomarker and then comparing that to the population.
Yeah, and that's a good question. As I mentioned, the speed for bioanalytical technology is pretty fast, so that's the data generation step. Well, when the data are generated, these are, you can think of them equivalent to imaging data, the mass spectrometry spectral images. And these data are pretty big, and the order of hundreds of gigabytes or per 100 samples. So, and all of this data, we have to do some processing on premise and upload to the AWS cloud, and then using the distributed computing platform that Edmond and the team built to do the machine learning-based extraction. The challenge is really to look at each of these peaks. As mentioned, there are millions of these peaks, but only about 10,000 are true signals. So we really have to go through each one of them and filter out the noise and keep the signal, and then do the downstream statistical and machine learning analysis. So the production processing would take, depends, so we're trying to scale it as much as possible because many of these can be processed in parallel. And it typically uses thousands of core hours to process one set of data. And the downstream analysis is just more understanding the clinical trial design and the clinical biological questions being asked, and then do the statistical and the modeling based on that. So that time can vary from days to weeks.
Thank you very much for the talk. I want to ask you, in terms of data engineering, processing, machine learning, what was one of the big challenges you had, and how did you overcome using the platform?
Sure. Thank you for the question. I think Edmondo would be better at answering this. I will try. So the biggest challenge here, I think, is the automation process. So before we were able to use any machine learning method or any distributed computing, all of this data processing were literally done by human. And the current existing software cannot even load the data. It will just crash because any existing software in mass spectrometry data processing were never designed to handle this much amount of data. And so people in the lab had to manually look at the data and label these are the good peaks that we can use. And with the machine learning method and the cloud computing, we can really turn this months of tedious work time into just a day of AWS and Databricks time. So this really automate the process and make it more scalable and make the data turnaround much faster, so we can focus more on the biological insight instead of the tackling with all this data extraction.
As an organization, do you try to work with generalists that know both the clinical aspects as well as the machine learning, or do you have dedicated data science teams which have no idea about what they're doing, consulting folks from the medical or biological field and sort of bridge the gap there?
Yeah, that's a good question. So we're a very interdisciplinary team. We have people with a clinical background, with analytical chemistry background for operating the machines, and we also have a data scientist with biological background, as well as a really excellent data engineer and architects like Edmondo's team that focusing on the software engineering and data infrastructure. But we try to learn from each other because the best way to tackle the problem is to understand the context and be able to speak other teams' language. So I think people came in with different background, but it's really through working together, and we learn each other's language and learn how to work with each other and utilize everyone's strengths.
Thank you for the presentation. Quick question about clean rooms and probably the opportunity with all the amount of data you have that could be joined with other either public data sets or even private data sets where people may not want to hand over the data set, but possibly you can join the queries and run the queries in a clean room. Are you doing that type of work with other organizations right now? And if not, sort of, is that something that you all are thinking of standing up and some of the complexities associated with that?
Yeah, that's a good question. So we're a company for business purpose, but really want to promote human health and discovery. So we are very willing to share data in any way, and we are also an academic spin-out, so for the research purpose as well. We actually try to integrate all the data that's publicly available, like all the genome-wide association studies, summary statistics, and all the microbiome data that's publicly available with the sequencing and the phenotypic data, and together with the data that we generated. And we also share out our summary results because just by compliance reason, we cannot share individual level data, but we share our summary results and the research discoveries with other research institutions and academic groups. And so we want to keep moving this process, and maybe since the company is still pretty new, it's only 1 year old, we're trying to develop a relationship, a partnership, and to get more and more data, especially the NIH genomics data and some other large national studies or large clinical trial repositories to really provide our technology as another dimension of a big omics data, and together with the existing data format as mentioned, other somatic mutation or any type of existing big data, all together to push the frontier of the discovery.
Thank you very much. Please give a big round of applause to our speaker. To remind